Hey everyone, and welcome to another video here at Whiteboard Doctor. Thanks for joining us today. Quick disclaimer before we move on. None of these videos are intended to be acted upon as medical advice. Please pause the video here and read the disclaimer in its entirety before moving on. Channel plug. Here at Whiteboard Doctor, our mission is to bring you interesting, relevant, and understandable medical education for all types of lifelong learners, trainees, and practitioners. If you want to follow along, we do have a lovely subscribe button in the bottom right-hand corner of all the videos. Don't forget to hit that like button. And lastly, if you'd like to support us outside of viewing our videos, we have several ways in which you can do that, linked in the video description and pinned comment. Stay well, keep learning, and back to the video. Alright, and welcome back to another video. Today we're going to dive into a topic that is, near and dear to our hearts, something that we feel is very, very important, and something that isn't talked about all that much. What if we told you that there was something that was estimated to cause as many deaths as cancer by 2050, and that there hadn't been any new treatment discovered, any new class of treatments discovered since 1987. It's underfunded, underfocused, and that is antibiotic resistance. So the title of this video is Antibiotic Resistance, a Global Catastrophe ? We work in the hospital, in the ICUs, in the emergency departments, and the floors, and this is something that we come across every single day, especially in the ICUs. Bacteria that have such significant resistance to our current antibiotics that sometimes there's no antibiotics to treat, sometimes there's just one or two, and those antibiotics tend to be things that are very toxic. There's studies upon studies upon studies showing that antibiotic resistance is becoming a very, very significant issue. 1.29 million deaths in 2019 are attributed to antibiotic-resistant bacteria. Up to 10 million deaths by 2050 are estimated, and that's as many deaths as cancer is causing. There's actually been a 35-year discovery void in terms of new antibiotic classes. 35 years, that's massive. And it is underfunded and underfocused. So that's what we're going to focus on today, is these superbugs, some call them, these bacteria that are resistant to many, many antibiotics. So to start, a little bit of history. In 1928, so almost 100 years ago, this gentleman here, his name is Alexander Fleming, all right, he took a 2-week vacation to Scotland. He worked in London in a lab, and he decided to take a vacation. Well, lucky for us, Alexander Fleming was a little bit, nonchalant in his cleanup, and he left, petri dishes, agar dishes, with bacteria growing on them. That bacteria was Staphylococcus aureus, and he left them in the sink. He didn't clean them up. He didn't throw them away. When he returned, he noticed that there was some fungi growing on the petri dish, and that's what this circle is. This circle is a petri dish, and he noticed that the bacteria, in red here, weren't able to grow close to the fungi in orange. And he was very astute, and he said, that's funny, because that bacteria should be growing all over that agar plate, so why can the bacteria not grow close to the fungi? And he tested that and discovered that the fungi was secreting what we now call antibiotics, in this case penicillin. And that was the penicillium fungi. That's why we call it penicillin. And that was the discovery, actually, of modern-day antibiotics. Alexander Fleming, and this is a picture from this Times article linked in the video description called How Being a Slob Helped Alexander Fleming Discover Penicillin. Some strong verbiage there in their title, but nonetheless, it was, a happy mistake. It took a very intelligent man, being Alexander Fleming, to recognize what was going on, but the reason it was going on is because he quite literally didn't clean up after himself before he went on vacation back in 1928. So antibiotics were discovered, a long, long time ago. The problem we're running into right now is that of antibiotic resistance. Antibiotic, often abbreviated ABX in the clinical arena. Antibiotic resistance. The World Health Organization actually deems it one of the greatest threats in our time. They go to say antibiotic resistance is one of the biggest threats to global health, food security, and development today. It can affect anyone of any age in any country. This is not just strictly, a problem in poorer countries. And it occurs naturally, but also misuse of antibiotics in humans and animals is accelerating the process. It's not just the World Health Organization, though. The Centers for Disease Control and Prevention in the United States talk about more than 2.8 million antibiotic-resistant infections every year in the U.S. alone. 35,000 people die as a result each year, according to this 2019 report. All right? And, if you count in things like C. diff infections, it is as high as 48,000 deaths. It has the potential to affect people at any stage of life, as well as healthcare, veterinarian, and agricultural industries. It makes it one of the world's most urgent public health problems. So we want to pause here, right? The World Health Organization, the Center for Disease Control, we, feel like there's probably identical statements in other countries and continents, healthcare organizations. But how many of you, let us know in the comments, how many of you have heard or learned about this problem, antibiotic-resistant bacteria and infections? Because, we're in medicine, so we talk about it, hear about it, think about it, but we feel many out there probably haven't even heard about it, let alone learned much about it or thought about why it's going on. It's a very under-recognized and underappreciated problem. It's one of those things, we see this over and over again for different problems, not just in medicine, but in life, is that, ignoring the problem until it is a real problem is kind of the path of least resistance, the quote-unquote easiest path. So we're kind of just barely getting by right now as there's more and more antibiotic-resistant infections, and we're kind of half ignoring it or not investing as much time and money as we should. But at some point, we're going to reach a tipping point where this becomes a massive problem. And at that point, we're going to be swimming upstream because we're going to be behind in discovery and behind in coming up with solutions to this problem. Why are we talking about this now? Well, it came to mind now because we at Whiteboard Doctor were just reviewing this global research on antimicrobial resistance, GRAM, a study they put out. And the study they looked at actually 204 countries and territories and antimicrobial resistance in these countries and territories. They acknowledge the data is limited for low-income countries. But what they found is highlighted here. This is a quote from their paper. I'll obviously link this in the video description. But after analyzing the data, they found that there's 1.27 million deaths across the globe could be contributed to antimicrobial resistance. And that was in 2019, 1.27 million deaths. Think about things that cause that degree of death each year across the globe. And again, we're not really talking about this. And if you just look at kind of deaths associated rather than attributed, so associated to antimicrobial resistance rather than being the sole reason for it, that number jumps to almost 5 million deaths globally. That's a massive, massive number, right? So this is huge, hugely important, a hugely significant, effect on global populations already in 2019. And that's why this popped into our head at Whiteboard Doctor, and we wanted to cover it for you because we think it's important for you all to know, and we think it's just going to become more and more important as time goes on. This study looked at the different types of infections that are most likely to be antimicrobial resistant, and they're common things. Respiratory infections, things like pneumonia, bloodstream infections, which can be very, very serious, and intra-abdominal infections. These are the three most likely infections to have antimicrobial resistance. All right. And when they looked at the actual bacteria that were most likely to be resistant, they're also common bacteria, things like E. coli, Staphylococcus aureus, streptococcal pneumonia, the most common cause of pneumonia. And these three bacteria alone account for 3/4 of all these deaths, all these 1.27 million deaths globally from antimicrobial resistance. So these common bacteria that infect a lot of people, right? Staphylococcus aureus can cause skin infection, cellulitis. Streptococcal pneumonia can cause pneumonia. E. coli can cause a number of things, intra-abdominal infections in the urinary tract, etc., etc. So many of you probably have actually been infected by one of these bacteria in the past. Luckily, it was not an antimicrobial resistant strain of this bacteria, but three common infections And 3 common bacteria are causing a massive number of deaths from resistance that they've developed to our current antimicrobials. So, how do bacteria become resistant? Is a great question, and understanding that helps us understand why this is becoming such a tough problem. These are 2 images actually from the CDC, Centers for Disease Control, so CDC images. We'll link them in the video description, the links to them. This one on the left talks about kind of the general ways in which bacteria can become resistant. So some of them are obvious things, right? Germs, they're saying germs that a bacteria develop new cell processes that avoids needing the antibiotics target. So essentially, they develop, if that antibiotic targets, let's say enzyme that the bacteria is using, the bacteria says, meh, we'll make a new enzyme, so that antibiotic doesn't work anymore. Sometimes these bacteria change or destroy the antibiotics with certain enzymes. So that's brilliant, right? The bacteria goes, okay, you're going to give me this antibiotic. I'm going to produce an enzyme that breaks down that antibiotic and destroys it before it can kill me. Some bacteria change the entryways that the antibiotic would use to get in. So if that antibiotic needs to get inside the bacterium to have an effect, the bacterium says, all right, well, I'm going to develop a protein to put on my surface that prevents that antibiotic from getting inside of me, deflect it away, and thus that antibiotic can't affect it. Some bacteria change the antibiotic's target. So if the antibiotic is going to target an enzyme, they just change that enzyme so that the antibiotic can't attach to it anymore. And some bacterium just get rid of antibiotics by using pumps to pump them out. So they can break down the antibiotic, they can prevent the antibiotic from entering, or they could pump it back out. All these things, are really quite impressive. Obviously, we wish bacteria weren't able to develop these things, but there's a lot of tactics bacteria employ that are quite impressive. Pumping antibiotics back out of themselves, out of the bacterium, so that the antibiotic can't help or can't affect them. Destroying or breaking down that antibiotic once it gets inside of them. All these things are really impressive. And we wrote some antibiotics, for those of you in healthcare that are affected by this. For instance, destroying the antibiotics is like a carbapenemase. ACE meaning enzyme. Carbapenem is a type of antibiotic, so an enzyme that breaks down carbapenems. All right, those of you who work in healthcare have probably seen this. In fact, in the ICU that we are currently rounding in as we speak, we have a carbapenem-resistant bacteria infecting one of our patients. It's abbreviated CRE. And carbapenem is a really powerful antibiotic, so this is significant. Using pumps to pump out antibiotics, Pseudomonas aeruginosus, Pseudomonas uses that tactic often to be resistant to antibiotics like fluoroquinolones or trimethoprim sulfa, which is Bactrim. Even more impressive than these things, though, are the ability for bacteria to share their resistance with other bacteria. And that is what this image is showing. So essentially, bacteria can share their resistance with their kind of neighboring or other bacterium. And they can do this through 3 kind of different tactics. One is plasmids. These are circles of DNA that can just move between bacterium. Another one is transposons, which are small clumps of DNA. DNA is genetic material that kind of codes some of these resistance abilities. And these transposons are small pieces of DNA that can go into and change the DNA of a cell. And these can move from chromosome to plasmids and back. So, these resistant genes and transposons can go into plasmids and then back into the chromosomes of the bacterium itself. And the third is phages. And phages are viruses that actually attack bacteria, and they can carry DNA from one bacteria to another. So what this translates to is kind of down here, these 3 things: transduction, conjugation, and transformation. Transduction being genes are transferred from one bacterium to another through these phages. So a phage or a virus attacks the bacterium, replicates, accidentally sucks up some of these genes for antibiotic resistance, and then goes and infects another bacterium and injects that gene for antibiotic resistance into the new bacterium. And now that bacterium is also resistant to the antibiotics that first bacterium was. Conjugation is when these resistant genes are transferred between bacterium. So one bacterium interacts with another bacterium and they exchange their resistant genes. Another one is transformation, which is when resistant genes are released from nearby live or dead bacterium. And then a new bacterium comes across that genetic material floating out in, the world and sucks up that antibiotic resistant gene, so then it is now resistant to antibiotics. So these are, again, very impressive tactics, right? If one bacterium develops resistance, it can give, it's not like it's an intentional giving, but give that resistant gene to other bacteria that it's near. And then those bacteria can be resistant. So you can see how this is becoming such a big problem because these bacteria have these advanced tactics for evading our antibiotics. So mutations, as we've talked about in regards to COVID and viruses many times, they're a natural phenomenon, right? Mutations happen randomly. And the bacteria, when they mutate, again, they might mutate in the ways that make them resistant to antibiotics. But then they also, as we talked about in this picture, can share those resistant genes. So if one bacterium evades the Grim Reaper, being, that an antibiotic that we're using to treat that infection, if it evades that antibiotic and develops resistance, it then can share that wealth of resistance with, we said, their friends and family, aka other bacteria. And that circle goes round, round, and round. So why do bacteria become resistant, right? We talked about they can mutate and form these resistant genes. We've talked about what exactly that resistance might look like, whether they're pumping the antibiotics back out of the cell, breaking down and degrading the antibiotics, sharing those antibiotic-resistant genes with other bacteria. But what do? We as healthcare providers or as, farmers or the general public, what do we do to help breed resistance? There are a few things that are big problems. One is antibiotic overprescribing. So antibiotics treat bacterial infections only. That's it. They don't treat viral infections. They don't treat inflammation. There's some caveats to this. They don't treat fungal infections. They treat bacterial infections. So if you have pneumonia from a virus, antibiotics aren't helpful. If you have bronchitis, antibiotics aren't helpful. If you have pneumonia from a bacteria, you do need antibiotics. If you have a sore throat or pharyngitis from a virus, antibiotics aren't helpful. If you have pharyngitis from bacteria, they are. So we as healthcare providers overprescribe antibiotics. And as patients, often we see patients who request antibiotics and sometimes get very upset if we don't want to give them an antibiotic because it doesn't seem like there's infections from bacteria. But just know, every time you take an antibiotic, when you don't need one, you are breeding possible resistance to that antibiotic, both for yourself in the future and for the general population. We treat patients who sometimes need antibiotics multiple times in a year, and if you've received intravenous antibiotics, you are at much higher risk to have an antibiotic-resistant infection in the coming months. So they're not harmless. Next time your doctor, we wrote this here, next time your doctor doesn't prescribe you antibiotics, assuming it's not a bacterial infection, consider thanking them rather than getting upset because, again, they're not harmless, not for you or for society. Because you take them, you're at higher risk for antibiotic-resistant infections in the coming months. And then also, society's at higher risk for infections that are resistant to those antibiotics. Does that make sense? Hopefully it does. Let us know if it doesn't. Another thing is actually antibiotic underprescribing, and let us explain this. So this is not that providers should prescribe more antibiotics. It's that we need to make sure that if we're going to start antibiotics, that we fully treat the infection. Because if you only half treat it, if you give an antibiotic that is supposed to be taken for 7 days and you only prescribe it for 3 days, or if the patient is supposed to take it for 7 days and they only take it for 2 days, that infection might be incompletely treated, and as such, it gives the bacteria that survive the incomplete treatment more ability to replicate with resistance to whatever antibiotic you were taking. Whereas, if you completed the whole course and killed all those bacteria, then there wouldn't be any, ability to get resistance. So you have to make sure if you do have a bacterial infection, take antibiotics, that both the prescriber prescribes it for the right duration, the right number of days, and the person taking the antibiotics takes all of those antibiotics as prescribed. And then a third way, and this is really, really significant, is actually through livestock and farming. So what happens is that livestock and farming, a massive number of animals, cattle, chicken, pigs, get antibiotics. All right. In this report, through CIDRAP, which is the Center for Infectious Disease Research and Policy at the University of Minnesota, over 6 million kilograms of medically important, aka antibiotics that can be used for people, 6 million kilograms of medically important antibiotics were sold for use on farms. The breakdown for that was 41% were used in cattle, 41% were used in swine or pigs, 2% in chickens, 12% in turkeys, 4% in other animals. So we are using antibiotics in all of these farm animals, and that is also breeding antibiotic resistance. Down here it says the class of medically important antibiotics most frequently sold for use in livestock in 2020 were tetracyclines, penicillins, and macrolides. And just so people know, an example of a tetracycline is doxycycline. This is used to treat pneumonia out in the community. Many of you probably have taken it. It could be used to treat COPD exacerbations. It can be used to treat soft tissue infections, all in humans. But a ton of livestock are getting tetracyclines, and that can breed resistance to them. Macrolides are things like azithromycin. This is the commonly prescribed Z-Pak. Many of you probably have taken a Z-Pak for things like respiratory infections. And penicillins, there's tons of penicillins. Many of you are probably familiar with penicillins, amoxicillin, augmentin. All of these antibiotics being used in livestock are also important for humans. And now some livestock might need antibiotics, but there's a massive focus on the millions of kilograms of medically important antibiotics used on farms and the possible resistance patterns resulting because of that. So these three things are some of the causes of antibiotic and antimicrobial resistance. So it gets into our next thing. Okay, there's antimicrobial resistance, but we are a very smart society and world. Why don't we just create new antibiotics? Are we not able to? The question is no, we absolutely are able to. But like this article in Nature says, Big Pharma has abandoned antibiotics, right? This was published in Nature actually back in 2020. In 2017, it was estimated to cost $1.5 billion to produce a new antibiotic. This is just a single one new antibiotic. $1.5 billion. And a new antibiotic is only estimated to make, at most, $46 million a year in revenue. And this discordance is just not financially, there's no financial incentive to do that. So it's financially not promising for these big pharma companies to create new antibiotics because it costs so much to create them, but they won't make that much revenue. Some of the reasons for the lack of revenue is because antibiotics are priced or regulated in many countries so that they don't become obscenely, is that word we tried to write, expensive. And countries like the United Kingdom, Australia, Canada, etc. And we say this is rightfully so. We work in healthcare, we see the everyday challenge of people being able to afford their medication. So we're not saying antibiotics should be more expensive at all. But part of regulating them and pricing them at affordable prices creates this lack of yearly revenue, and then this feedback loop where big pharma doesn't want to invest the money to create new ones because they're not going to make much money. And that's lots of comments on just, healthcare in general and the problems with it. But that's one of the reasons. In addition to that, there's about typically a 5 to 10 year exclusivity period before generic versions can be produced. So if they produce a new antibiotic, only have 5 to 10 years to reap any, financial benefit before other companies can just put out generic versions of that antibiotic. And this is, becoming a vicious circle because as more antibiotic resistance occurs and there's fewer new antibiotics, prescribers are trying to save the new antibiotics so that we don't breed resistance to them. So we have something in our back pocket for those really, really resistant infections. And what that does is that this 5 to 10 exclusivity period starts to tick by. Prescribers aren't using the new antibiotic that much because we want to save it so that we don't have resistance to that new antibiotic. But then Big Pharma doesn't want to produce any new ones because they won't make enough money to recoup that initial investment. So obviously there's lots in here, comments on the political arena, society, healthcare, all that stuff. And we're not commenting on any of that in particular, but this is just to say that we are able to make new antibiotic classes, but there's not a financial incentive for people to invest a lot of money in doing it. This is kind of another testament to that. This is few antibiotics under development. This chart is taken from this REACT 15 years. We will link this article in the video description, but it's essentially a timeline, right? It says right here, timeline of the discovery of different antibiotic classes in clinical use, the discovery void, a period from 1987 until today. The last antibiotic class, the last new class of antibiotics was discovered in 1987, lipopeptides. And there's been a discovery void. There hasn't been any new class of antibiotic. Now, there's been some new antibiotics, but they're just kind of spin-offs of previous classes. There's not a brand new class of antibiotics since 1987. Think about that. That is obscene. That's wild. Wild. To kind of put it in perspective, we Googled. This is what we Googled. How many cancer drugs are approved each year, right? And, we didn't verify that this was true, but this website, after we Googled it, BeckersHospitalReview.com said there was 20 cancer drugs approved in 2020. The FDA then approved 53 new drugs this year in total. So we haven't had a single antibiotic since 1987, which is, 97, it's like 35-ish years. We might have done our math wrong, but about 35 years, whereas there's 20 cancer drugs approved in 2020 alone, right? So this is not a matter of ability. This is not a matter of not being able to discover new antibiotics. It is a matter of not prioritizing the discovery of new antibiotics because they're not financially beneficial to pharma companies that spend all this money to produce new drugs. These pharma companies would rather produce cancer drugs because they'd make more money off that than new antibiotics, for a lot of the reasons we talked about. All right. So what type of ways are out there to fight bacterial infections beyond just antibiotics? Well, there are some things people are working on, and these might not be big pharma, but kind of smaller labs and smaller companies. And the first is bacteriophages. So this might sound familiar. We talked about bacteriophages. We're going to zoom up when we were talking about how bacteria can share their genes for antimicrobial resistance, right? And that was here, bacteriophages, all the way on the right here. And bacteriophages are viruses that attack bacteria, all right? Just like viruses that attack humans, but these are specific to bacteria. So some people said, if there's viruses that just kill bacteria, why don't we see if we can hone those bacteriophages to kill infectious bacteria? And that's what people are studying. And this honestly has been talked about for many, many, many years. It seems like it's something that would be effective, again, if someone invested the time and money into it. But they've used it before. For instance, there was a 15-year-old boy who had a liver infection who was treated with bacteriophages successfully. And this is just a bacteria here with some cilia. This is the bacteriophage. It goes on the bacterial cell membrane and it injects its genetic code, and that, does what it does, and that bacteria ends up being destroyed, and that virus, that bacteriophage virus, just like a virus that infects humans, replicated and goes out and infects more and more bacteria that it can destroy. So it makes a lot of sense that this would be effective in treating infections. And people are actively developing and working on it, so it'll be interesting to see where that goes. Another big area is probiotics, and many of you probably have heard this. So probiotics are essentially good bacteria. We actually have more by some estimates, but at the very least just about the same number of bacteria in our body as human cells. tons in our gut, in our lungs, on our skin, all over our body. And these good bacteria actually prevent bad bacteria from replicating. And that's why when you get antibiotics, you're at risk for an infection in the gut called Clostridium difficile or C. diff, because even some good antibiotics kill good bacteria. And with less of those good bacteria, bad bacteria such as C. diff could replicate and start an infection. So researchers have said, maybe we could treat infections with probiotics, and they're studying mice right now using bacillus, a good bacteria, and they've had some success in treating infections such as MRSA methicillin-resistant Staphylococcus aureus infections with probiotics. And something we do, and it's now kind of in the gray because there's some challenges with it, but sometimes we treat Clostridium difficile infection with something called a fecal transplantation. Fecal being poop, transplantation being transplant, and you actually take poop from someone healthy and transplant it into the intestines of someone with a C. diff infection, and all the good bacteria in that healthy poop takes over and kicks out the bad C. diff bacteria to treat the infection. And many other discoveries of just different compounds, both naturally occurring and synthetic, that might be able to kill bad bacteria while not harming human cells and not harming good bacteria. So this is something we really wanted to talk about, given, how often in the ICUs and in the emergency department, someone has an infection. We take cultures, whether it's cultures from their lungs or from their blood, and it grows a bacteria. And we have them on powerful antibiotics, and, it usually takes a day or two to grow a bacteria, and then it takes a day or two for us to know what antibiotics that bacteria is susceptible to. And more, it seems like every year we're finding bacteria that are just resistant to so many powerful antibiotics and antimicrobials to the point where, there's only one or two antibiotics that might treat that infection. And as the resistance goes more and more, something we do, right, is what we want to use as first line, first line therapy. So first line therapy for an infection. We want that to be an effective antibiotic, antibiotic ABX, antibiotic, and then also non-toxic, right? So a lot of the first-line antibiotics we've been using have now resistance patterns. So now bacteria are resistant to them. So what do we have to do? Well, we still need an effective, I don't know why we can't say that, effective antibiotic. But what we have left are some of the antibiotics that we haven't been wanting to use because they're more toxic. So now we have to use some of these antibiotics that are more toxic and cause renal injury, liver injury, things like that, to treat the infection, because otherwise the patient would die because their infection is resistant to the better non-toxic antibiotics that we'd like to use first line. So now we're having to use, things like second, third, fourth line antibiotics, to treat these resistant infections, which have a worse tox and adverse effect profile. So we come across this in our our day-to-day work, at least every week, if not every day or every other day, and we wanted to share it with you. So let us know what thoughts, comments, questions you have down below. Let us know if this was an interesting video. This is kind of going to be in line with those videos on medical stories worth telling, which is a new playlist we started and linked on our homepage. So let us know what thoughts, comments, questions you have down below. Let us know if you enjoyed this type of video. Obviously, let us know if there's anything else you'd like us to cover. And as always, stay well, keep learning, and we will see you all next time.